Hi, my name's Alex, and today I'm going to do an overview of the cell cycle. This is important to know for all areas of molecular biology, particularly if we're studying genetics. I want to point out that multicellular organisms rely on cell division for renewal and growth, whereas unicellular organisms divide for reproduction. If we jump on the next slide, the main thing to emphasize is that there are a few stages, two in particular. So interphase and M phase. Interphase is the blue phase that we can see here, and M phase is the yellow here. A common misconception is that M phase is the entirety of the cell cycle, and it's not. So interphase contains the G1 phase, S phase, and G2 phase. In this section, the cell grows and the organelles within the cell duplicate. In S phase, DNA replication occurs, so it's split from the one organism, doubles, so that eventually when the cell divides, we've got two sets of DNA so that we can produce two offspring. In G2 phase, we've got the final growth and preparation for mitosis. So pretty much making sure that everything went well in S and G1 is what happens here so that then we can divide effectively. Before moving on to the M phase in a bit more detail, I want to go through interphase in more detail. Let's start with G1. As I said, the cell grows here and organelles duplicate. The main goal is that it grows properly and it's functioning normally. We want to synthesize new proteins and keep duplicating organelles. At this point, the DNA is still in its original form, so it has not been duplicated yet. There's a G1 checkpoint that occurs near the end to ensure that the conditions are suitable for DNA synthesis, so that everything went well. We don't want to duplicate DNA and the cell if the cell itself is not good. So that's what the G1 checkpoint is for. And I'll go into a little bit more detail with this in a later slide. In S phase, the main goal is DNA replication. The chromosomes become sister chromatid pairs. DNA content doubles. So we call that going from 2C to 4C, as we can see down here. Histones and proteins are made for new DNA and centrosomes duplicate. If we're not familiar with centrosomes, they're a cell structure that helps organize microtubules, especially during cell division. Each centrosome contains two centrioles in animal cells, which acts as the main microtubule organizing center. This is relevant because during mitosis, centrosomes move to opposite poles of the cell and can help form the spindle apparatus, which pulls these two sister chromatids apart so that each offspring get one. Okay, move on. In the G2 phase, this is final growth and preparation. The main goal here is quality control. Make sure that we've duplicated efficiently in S phase so that we have the all clear to do mitosis and cell division. So during this process, the cell checks for DNA errors and repairs any mistakes that may have been made. Organelles are polished and the centrosomes finish duplicating. Microtubules and energy stores are prepared for mitosis, as we said. As the DNA has already been duplicated, we remain at 4C. There's no more copying that happens here. And there's the G2 checkpoint that makes sure all of this is done well enough so that the cell can divide in the next phase. Some practice questions, and probably not the kind of questions that you'll find in the exam unless it's those one-off one-point questions that are kind of about uh, recall, not exactly application, but during which phase of interphase does the cell replicate its DNA? If you've been paying attention, you'll know that that's B, S. Which of the following correctly describes the G2 phase? So what did we say happens in G2? It's not A, that's G1. B is S phase. C, cell checks for DNA damage and prepares for mitosis. Probably. D, sister chromatids are separated. No, that happens in mitosis. So it's C, okay? What happens to the DNA content during S phase? We said before that it duplicates, and if it starts off as 2C, it becomes 4C. So the correct answer is B. Which structure duplicates during interphase to organize the mitotic spindle? We said B, centrosomes. Okay. Now let's move on to M phase or cell division. This is probably best understood when it's visualized, but I will do both just in case visualization is not your learning style, and you need it. I mean, it's good to have it both explained as well. So we'll go through both and then hopefully the two will combine to stick and make sense, okay? This can be divided into mitosis and cytokinesis. So mitosis is where the nucleus and DNA content divides, whereas cytokinesis is where the cytoplasm separates to produce the two offspring. So all the way up until cytokinesis, it's one cell with double the amount of genetic content, and then in cytokinesis, this separates to have two identical offspring. A good acronym for memorizing the stages of mitosis is PMET, prophase, metaphase, anaphase, and telophase. I've got some brief descriptions that you can note down of each, but we'll go into a bit more detail in the visual here. In prophase, as you can see, chromosomes condense, the spindle forms, and the nuclear envelope breaks down. In metaphase, the chromosomes line up at the equator, and the equator is the middle of the cell. Anaphase is distinctly remembered by the separation of the sister chromatids, and it's got this distinct image of the separation that's pretty easy to recognize. So anaphase is probably going to be the easiest one to identify if given an image. I mean, so is metaphase if they're lined up along the equator, but make sure to be able to distinguish between anaphase where they're separating and metaphase where they're just lining up. In telophase, the nuclear envelope reforms and chromosomes decondense. So they're no longer in this formation, they decondense here. And then in cytokinesis, the two separate. Sorry, the one cell separates into two. Let's go into a little bit more detail in each phase. I'll explain why everything happens so that it's a bit more conceivable. So chromatin condenses into visible chromosomes. This happens because tightly packed DNA is easier to move without breaking, right? It prevents tangling and it allows accurate separation of sister chromatids later in anaphase. Okay. Each chromosome consists of two sister chromatids. This is because DNA was replicated in S phase, as we spoke about, and having identical copies ensures that each daughter cell receives a complete and identical set of genetic information, because remember, the end product is two identical daughter cells. The nuclear membrane begins to break down. Why do we need the nuclear membrane to break down? Because this allows spindle fibers to access the chromosomes, because if the membranes actually if it stayed intact, the microtubules couldn't attach to the chromatids, and then therefore they wouldn't be able to separate into the adjacent daughter cells. The nucleolus disappears. Why? Because ribosome production pauses and RNA transcription stops during mitosis. So the nucleolus disassembles as the cell reallocates resources towards division. Because we're going to pause transcription. Our main focus during mitosis is division. Centrosomes move to opposite sides of the pole, and this positions them to form the mitotic spindle and establishes the axis along which the chromatids will later be pulled apart. Okay. Finally, the mitotic spindle begins to form, so microtubules grow out from each centrosome, creating the structure that will attach to chromosomes and separate the sister chromatids, right? Without this, the chromosomes couldn't be distributed evenly to the daughter cells later. In metaphase, the reason that the chromosomes line up along metaphase, this alignment ensures that each sister chromatid is positioned so that it can be separated evenly towards opposite sides of the cell, preventing unequal DNA distribution, because that would result in things like aneuploidy, and the integrity of the two daughter cells wouldn't be great if it wasn't distributed evenly. So I think you can kind of pick up like a really fundamental part, especially of these initial parts of mitosis, is even distribution to each of the daughter cells. So that's why we have the lining up. The spindle fibers or the microtubules will attach to chromosomes at their kinetochores. This attachment provides the physical link needed for pulling the chromatids apart. Without the kinetochore attachment, the spindle couldn't generate force on the chromosomes. Tension is applied across sister chromatids from opposite spindle poles, and this tension acts as a mechanical check to confirm that each sister chromatid is connected to the correct pole. Proper tension is required to avoid chromosome missegregation. Okay. Finally, the cell performs the metaphase checkpoint. So the cell will ensure that chromosomes are properly attached to the spindle and that the tension is balanced across each of the chromosomes. The checkpoint prevents the cell from entering anaphase until everything is correctly aligned, reducing the risk of aneuploidy. So this is something that will probably, it's high potential that something like this can be asked on an exam question. What's the point of the checkpoint during metaphase? Okay, and you would say it prevents the cell from entering anaphase to ensure that everything is, prevents the cell from entering anaphase unless everything is correctly aligned. Why? To reduce the risk of aneuploidy or incorrect chromosome number in the daughter cells. Moving on to anaphase, the sister chromatids separate and are pulled towards opposite ends of the cell. This happens because cohesin proteins holding the chromatids together are cleaved, allowing each chromatid to move independently, so each daughter cell receives one copy of each chromosome. Sorry. um, Bit of repetition here. The amount of times I've said so that each daughter cell receives an equal number of DNA and equal content in each daughter cell is created. It's so that it drills in. Obviously, when something is repeated, note that it's important and it's likely to be assessed. Spindle fibers shorten as microtubules depolymerize. This shortening creates the pulling force that moves chromatids towards the centrosomes at opposite poles, ensuring accurate chromosome segregation. The cell elongates as non-kinetical microtubules push against each other, and this elongation increases the distance between the two poles of the cell, helping physically separate the forming daughter cells and setting up cytokinesis. Finally, the separation of chromatids marks the transition from chromatids to chromosomes. This distinction is important because once they're separated, each chromatid is considered an independent chromosome in its respective daughter cell. And we move on to telophase. Here, chromosomes begin to decondense back into chromatin. And what this does is it makes DNA accessible for transcription and normal cell functions once division is complete. Chromosomes will reach opposite ends of the cell, and this happens, you can probably tell me, so that each daughter cell will receive a complete and identical set of chromosomes. The nuclear membrane reforms around each set of chromosomes, and this restores two separate nuclei to protect the genetic material and preparing the cell for cytokinesis. You can probably put this one together by yourself from previous knowledge in biology and considering the function of the nuclear membrane itself. Next, the nucleolus reappears in each nucleus, and this resumes ribosome production, so it allows the daughter cells to carry out protein synthesis and normal cellular activity on their own. Finally, the mitotic spindle disassembles, and this is because the microtubules are no longer needed for pulling the chromosomes apart, and their components can be recycled for other cellular functions. So let's do a practice, and based on the quick run through, finally, cytokinesis. So this isn't part of mitosis, this is separation afterwards, but in cytokinesis, the cytoplasm divides to form two separate daughter cells. This happens so that each new cell has its own complete set of organelles and enough cytoplasmic material to function independently. A contractile ring of actin and myosin. A contractile ring of actin and myosin filaments will form around the cell's equator. So this ring pinches the cell membrane inward, creating a cleavage furrow that eventually splits the cell into two. So that picture that I've got there is at the end of cytokinesis, but the cleavage furrow I'm talking about is what's going on here. Okay, so telophase will kind of end in that position, and then we can say, all right, yep, we're in cytokinesis. And then cytokinesis will be the process of continuing to break down the cleavage furrow until this is the end point, the two daughter cells. As I said, the cleavage furrow will deepen until the cell is fully divided. This ensures that the cytoplasm, organelles, and cell membrane are evenly distributed between the two daughter cells. After cytokinesis, each daughter cell enters the G1 phase of interphase, so it starts all over again. And this allows cells to grow, carry out normal functions, and prepare for the next cell cycle. This is continuous in most cells, not in all. For example, neurons do not continuously go through the cell cycle, but in the ones that do, this is how it happens. Let's see if we can identify which phase of M phase each of these are in. Have a go by yourself. I'd say the best place to start is these two. We said that they're probably the most easily recognizable. If the chromosomes are lined up along the cell equator and are still intact, it's metaphase. If they're beginning to be pulled apart, the sister chromatids are beginning to be separated, anaphase. Before this happens, the cell looks like this, so that's probably interphase. And then these two, the cleavage furrow is distinct of the end of telophase. And if it's finally separated, that is an indication of the end of cytokinesis, which is the division of the two daughter cells. Sorry, the division of one cell into two daughter cells. Okay, if you can do that little exercise, you're doing pretty well. If not, it's okay. Go through the material again, maybe in a different way. Watch a few videos. There are a lot of animations on things like on YouTube and stuff. And the more you expose yourself to it, the more easily recognizable the cell phases, the stages will be. Okay. Let's do some practice questions. Again, these are more recall just to build up your confidence a little bit. I would recommend doing some application questions as part of your revision and study before an exam on this content. But just so that we've got a good foundational understanding, during which stage of mitosis do chromosomes line up at the cell's equator? What do we think? They're lined up in both B and C. However, the question here is not where the chromosomes line up and separate, it's just line up. So I would always distinguish these two by whether or not they're being separated. If separation is not mentioned, the correct answer is B, metaphase. Sister chromatids separate and move along opposite poles during, that's what we just discussed, anaphase. The nuclear envelope begins to reform around chromosomes during. When is the nuclear envelope broken down? That would be in prophase. Okay. And then they'd be lining up in metaphase and anaphase. So it would begin to reform in telophase. The correct answer there is D. Which structure forms in animal cells to physically divide the cytoplasm during cytokinesis? If you can identify that this is the cleavage furrow, that's correct. These two are not indications of division, and this one is for plant cells, okay? Which of the following is true about the mitotic spindle? It forms during G1 phase, it's composed of actin filaments, it attaches to kinetochores and pulls chromatids apart, it disassembles during S phase. I believe we mentioned that the answer is C. It attaches to kinetochores and pulls the chromatids apart. That's a very key part of mitosis. Explain how errors in M phase can lead to aneuploidy and relate this to human diseases. So what happens in M phase? We want to distribute, we've repeated it lots of times, we want to evenly distribute DNA contents between the two daughter cells. If they're not evenly distributed, then the two daughter cells, one will probably have not enough chromosomes and one will have too many. For example, in Down syndrome, we call that trisomy 21, where there is an extra chromosome in the 21st pair. So that's how you relate that to human diseases and explain how it leads to aneuploidy. Next question, compare metaphase and anaphase in terms of chromosome movement and spindle function. So you can say something like, when asked a comparison question, I think it's good to include similarities and differences, at least one key one for each. So similarity and difference, you can say, in both metaphase and anaphase, in terms of chromosome movement, the chromosomes line up towards the cell at the cell's equator. However, in metaphase, they are not separated, whereas in anaphase, the mitotic spindle will pull the sister chromatids apart. That's perfect. Okay. All right. Now we also have to consider G0 phase. This is a resting or non-dividing state that a cell enters after G1 when it's not preparing to divide. So a good analogy is a work schedule where G0 is like taking time off or acquiring, okay? You can take temporary leave like quiescent cells. Permanent retirement refers to neurons, cardiac cells, and red blood cells. They are not constantly dividing. They stay in G0 in resting phase. Some cells have forced retirement due to damage. Okay, these are called senescent cells. A little bit more detail. The cell exists in an active cell cycle here, so no DNA replication or division occurs. Metabolic functions do continue normally, and it can be temporary or permanent depending on the cell type. So those are the ones that we just went through. The purpose of this is that it saves energy by not constantly dividing. It allows specialization, for example, into neurons, and it prevents damaged cells from dividing, which is called tumor suppression. Okay. Now, the checkpoints, as we alluded to early in the slides, these are very important for linking biology with health and disease. If they did not exist, we would continue from interphase to synthesis, even if interphase didn't go well. And what would happen if interphase didn't go well? Well, the cell wouldn't have grown properly, it wouldn't have good nutrients, and the DNA might be damaged. So then we're going to go into S phase and duplicate damaged DNA. And then if this isn't regulated, we're going to go into G2 and it's going to say, yeah, all good, because the checkpoint for G2 is actually supposed to say, hey, actually, this is not right, and we don't want to divide this cell and take it into mitosis and cytokinesis. But if the G2 checkpoint doesn't work, then that'll just go ahead anyway, right? And then we're going to have division of a cell with damaged DNA, okay? And this is how things like cancer are caused. Because of the checkpoints, if something is wrong, the cell can pause, repair, or trigger apoptosis, which means programmed cell death. So to go into a little bit more detail, I just explained it like pretty casually there, but let's go through in a little bit more detail. Whoops. During the G1 checkpoint, this is called the restriction point, and it determines if the cell is ready to enter S phase, aka if it's ready to do DNA synthesis. So the questions that it's asking is, how's the size? Is the cell big enough to divide? Are there sufficient nutrients, sufficient energy, and raw materials there for us to go through this process? Are there necessary growth factors? And is the integrity of the existing DNA good enough for replication? If the conditions are not met, the cell might go into G0 phase. This is regulated mainly by something called P53 and there's a cyclin CDK complexes, which we'll go through at the end. But that's the G1 checkpoint. G2 determines if the cell is ready to enter M phase or mitosis. So after S phase, was the DNA replication done well? Is it fully and accurately copied? How's the integrity of the DNA? It must ensure that there is no damage that has occurred during replication. And how's the cell size? Does it have enough resources and is it big enough to be divided and to be given the all clear to go through mitosis? If DNA, my mouse is so sensitive. Where was I up to? I think here, G2. If DNA is damaged or incomplete, the cell cycle is halted by the G2 checkpoint until it's repaired. This is controlled by another cyclin CDK complex and DNA damage sensors. The M checkpoint is also referred to as the spindle assembly checkpoint. So this occurs during metaphase of mitosis, and it checks that the chromosomes are all attached to the spindle fibers via the kinetochores, and that the chromosomes are properly aligned to ensure equal segregation to daughter cells. If errors are detected, then anaphase is delayed until the chromosomes are correctly attached so that when it separates, each daughter cell receives good DNA and an equal amount of it. This prevents aneuploidy, abnormal chromosome number, which we spoke about in the Down syndrome example. Okay. So a little bit about the CDK, cyclin-dependent kinases. These are a family of enzymes that control the cell cycle. They're called cyclin dependent because they only are active when they bind to proteins called cyclins. And what these do is they add a phosphate group or do phosphorylation. They bind to specific target proteins and they change these proteins' activity. What this does is it acts like a molecular switch that tells the cell to either start copying DNA, prepare for division, or to divide now. So you can see here that there are different cyclin CDK complexes at each different phase of the cell cycle, which do different functions. Okay. That's all I've got for today in the cell cycle, just a brief overview. I hope it's been beneficial. As always, if there's a key point that stood out to you that was confusing, I recommend focusing on that during study first. Do not just do not do repetition over the parts that were most straightforward. And focus during exam preparation on application questions. Okay, recall questions are good initially to make sure that your foundational knowledge is good and you're following along in the lectures, but really focus on application questions during exam revision because those are likely the ones that will appear on the final exam. Okay, thank you. See ya.